Viruses are responsible for one of the last great unconquered areas of infectious diseases. Now, these viruses come in a great variety of sizes and shapes. They actually range from about 10 millimicrons to about 300 millimicrons. Since there are chemical molecules, certain protein molecules, that are around 30 millimicrons in size, the viruses actually overlap with chemical molecules at one end. Now, at the other end of the scale, where we have living organisms, there are certain living organisms that are actually smaller than some of the larger viruses, such as vaccinia virus, for example. Now, this range between the molecules of the chemist and the organisms of the biologist, one could literally call a twilight zone of life. And this zone is actually filled in by the viruses. Now, it's interesting that some of the viruses can be obtained in the form of crystals. We obtained poliomyelitis virus in crystalline form just a few years ago. And if you take a crystal of poliomyelitis virus and slice it in half, such as you would slice an apple or an orange with a knife, and look at the slice of the crystal, this is what you would see. This is an electron micrograph of a section of a crystal of poliomyelitis virus. Now, each one of those little round objects that you see there represents an individual particle of poliomyelitis virus. This micrograph is at a magnification of about 1,000,000 times. Now, if you take one of those particles and use all the knowledge that we have about its internal composition and structure and make a model of it, you would see something like this. This model is actually at an enlargement of about 65 million times. You can see here it has protein subunits on the exterior, and we know that on the inside of this we have nucleic acid, which is the infectious material of the virus. Now, this is an example of a virus that causes disease in animals or in man. It represents one great class of the viruses. Now, there's another class of viruses that is represented by the viruses that attack plants, the so-called plant viruses. And here we have a model of a plant virus with the same magnification, about 65 million times. Except here, this is only part of it. If it were all here, it would be about 60 feet tall. And here again, you can see that it's made up of protein subunits on the exterior. And then inside of this virus, inside of the virus, you see the nucleic acid, which goes round and round internally, just forming sort of a spiral staircase. Chemists have learned much about the structure of these viruses, and this has provided us with much information concerning their nature. The other great class of viruses is the bacterial viruses. And here you see a drawing of a bacterial virus with a hexagonal head, the nucleic acid on the interior. One of my colleagues, Gunther Stent, is a specialist in this particular area, and I'd like to have him tell you something more about the bacterial viruses.
Bacterial viruses, that is, viruses which infect, grow on, and kill bacterial germs, were discovered independently in London and Paris during the First World War. These viruses have come to be known under the name of bacteriophages, a word derived from the Greek meaning eater of bacteria. Phages derived from the word phagein, to eat, and this means bacteria. Now, the discovery of viruses that infect bacteria, themselves the smallest organisms known to man, caused a tremendous excitement in the world of medical microbiology, because it was hoped that by means of these bacterial viruses, it would be possible to control and to eradicate all those bacterial diseases which had been the plague of mankind since the dawn of history. Now, the idea of this plan was a simple one. Let us take the case of a bacterial disease, such as cholera, caused by the cholera germs. It would only be necessary to isolate in nature the virus which infects and kills the cholera bacteria, cultivate this virus in the laboratory, and then disseminate it throughout the world. The anti-cholera viruses would then seek out the cholera bacteria wherever they can be found and destroy them, and thus rid mankind of the cholera threat. Now, this hope initiated a tremendous amount of work on bacterial viruses during the 1920s and 30s, with the intention of making a practical go of this bacterial virus therapy. But unfortunately, this hope proved in vain, because by 1939, it had still been impossible to cure or control any single bacterial disease by means of these bacterial viruses. However, during all that time, it was learned, the fundamental nature of the growth of these viruses were learned, and it came to be realized that they could be used as an excellent model system for the study of biological self-duplication. Now, the idea is the following. A bacterium is infected by a bacterial virus particle. Half an hour later, this bacterium bursts open and liberates into the world 300 virus particles, which are exactly identical to the parent virus which caused the infection. And so the problem that we can put ourselves is this. What has happened in that infected bacterial cell during the 30 minutes, during which the parent virus gave rise to 300 offspring? And it is in the hope of learning something about the detailed chemical and physical mechanisms which intervene during those crucial 30 minutes that much of the work during the 1940s and 1950s has been devoted to the study of bacterial virus reproduction. Now let's see, first of all, what such a bacterial virus looks like. Here we have a painting of a typical bacterial virus. At the tip of the tail, there are some tail fibers, and then the tail extends towards the head of the virus. The tail itself has a hollow tube in the middle, and the head is filled with nucleic acid. These viruses can be photographed in the electron microscope, and here we have an electron microphotograph of a particular bacterial virus, the virus T2, which infects the coli bacteria inhabiting the human intestine. You can see the heads and you can see the tails quite clearly. The size of this organism is about 1/100,000th of an inch, which will give you an idea of the magnification of this picture. Now, in order to study the chemistry and physics of reproduction of this particular organism, it's necessary to know its chemical composition. And here we're lucky because the composition is rather simple. It consists only of two components. One is protein and the other is deoxyribonucleic acid. Or DNA. Now, the actual chemical constitution of this substance is rather complicated, as complicated as its name. Nucleic, because it's found in the nuclei of all living cells. Acid, because it's acidic. And deoxyribo, because it contains the sugar deoxyribose. Now, the protein of the virus is the matter from which the tail of the virus is formed. And it is also forms a membrane, an outer membrane of the head. And the DNA is inside that head. Now, let's look at what the Bacterium coli, the host organism of this virus, looks like. Now, this sausage-shaped object here is the Bacterium coli, the host organism of this particular virus, T2. The tadpole-shaped objects here, the little tadpoles, are once more the T2 virus particles. This picture gives you an impression of the relative size of the two objects, host cell and virus. You can see that the bacterium is about 1,000 times larger than the virus which it infects. Well, we're now ready to consider the life cycle of this particular virus, the way this bacteriophage T2 reproduces itself. Now, here is a cutout model of the bacterial cell once more, the colibacterium. And here we have a model of the T2 virus particle. Now, the virus first attaches itself to the surface of the bacterial cell by means of the tail fibers, which anchor the particle to the cell surface. Then this tail of the virus contracts and injects some kind of a hypodermic needle into the interior of the cell. And then the DNA fibers, which are in the head, get injected into the interior of the bacterial cell. Now, once the bacterial DNA is inside, the viral DNA is inside the bacterial cell, it begins to reproduce itself several hundredfold in order to generate the DNA parts for the offspring viruses. At the same time, the DNA induces or presides over the construction of protein replicas of the virus particle which caused the infection. Some 10 minutes after the infection started, the first DNA replicas begin to condense with the protein replicas of the parent virus to constitute the structurally intact virus particles of the offspring generation. As time progresses, more and more such virus particles are formed, more and more DNA units condensed with more and more protein units until several hundred offspring virus particles have been constructed inside that cell. At about the 30th minute after infection, about 300 virus particles are in the cell. The cell bursts open and liberates into the medium the virus particles of the offspring generation. The virus offspring then look for other bacterial cells to infect and to repeat this reproductive drama. Now let's run through this cycle of events once more by means of a trick film which has been prepared for this purpose. Now here's the bacterial cell floating in a medium, and soon a virus particle diffuses into its neighborhood. It collides with the cell and finally fixes itself to the cell surface by means of the tail fibers. The tail now contracts and injects this hypodermic syringe into the interior of the cell, and the DNA fibers enter the interior of the cell. The virus reproduction is now ready to begin. The DNA fiber is somewhere in the interior of the cell, and the cell is getting ready to reproduce the fiber. At first we have one fiber, and then we have two fibers, and the two fibers each reproduce itself to give 4 DNA replicas exactly like the DNA of the parent virus. At about the 10th minute, we see a protein unit condensing around the DNA, and the first structurally intact virus particle has made its appearance. As time progresses, more and more such condensates take place, more and more virus particles appear inside the cell, and the whole cell fills up with hundreds of these offspring viruses. Finally, at the 30th minute, the membrane of the cell wall, ruptures, the cell explodes and liberates into the medium the several hundred virus particles which are ready to repeat this reproductive process. Now let's consider how we actually count the number of virus particles which we may have in a given sample. Now for this purpose, we take such a glass dish which is filled with a nutrient jelly. On the surface of this nutrient jelly, we place about 1 million bacteria of the type, in our case E. coli, Escherichia coli, of the type that is sensitive to the action of the virus which we wish to count. And at the same time, we place on the surface of this agar, of this nutrient jelly, a few hundred virus particles. This plate, containing the bacteria and the viruses, is then placed in an incubator at 100 degrees Fahrenheit and incubated overnight. Now, in the course of this incubation, the bacteria begin to divide and grow and cover the nutrient jelly surface with a thick carpet of bacterial growth. But as the bacterial carpet is formed, holes begin to develop in that bacterial carpet, holes which we call plaques. Now, these plaques or holes grow in size as the bacterial growth proceeds, covering the surface of the jelly with an ever thicker carpet. Now, the next day, when we take this plate out of the incubator and look at it, it will look something like this. You see the dense bacterial growth, and you see the plaques or holes which are in this carpet. Now, what is the meaning of these holes or plaques that we've seen develop on the surface of this nutrient jelly? Each place on the nutrient jelly where one of the virus particles had come to rest, it will find some bacterial cell in its neighborhood. The virus will then infect that bacterial cell, reproduce on it, and half an hour later, several hundred offspring of that virus will issue. These offspring will then infect neighboring bacterial cells and repeat the reproductive cycle. Their offspring will in turn infect neighboring cells, and so more and more viruses will be produced in that original focus, in that place where the virus particle, the parent virus particle, had come to rest. So as the bacteria grow and cover the nutrient jelly with a thick carpet, so there will grow a hole in the carpet which represents the virus colony descended from the original virus particle which we had placed on the nutrient jelly. The hole, therefore, represents an absence of bacteria which have been destroyed by the growth of the descendants of the original virus particle. Now, since each virus particle will cause the production of such a plaque or hole, the number of holes, finally, in the carpet will be equal to the number of virus particles which we placed on the plate, and therefore give us an indication, give us an assay of the number of virus particles which were present in our sample. Now, as we look at thousands of such plaques, as every good bacterial virus worker does every day, we find from time to time an unusual or variant plaque which looks different from all the rest. Now, here on this plate, there is one plaque which does look different from the rest. You see, this plaque here is larger than the others. Most of the plaques are small, whereas this one, this variant plaque, is large. Therefore, we call it a large plaque type. Now, if we pick the virus colony which is present in this hole or plaque and put a sample of these viruses on the nutrient jelly, then we find that all of the viruses which were present in this variant large plaque now themselves make large plaques. And here we see a close-up of the offspring viruses making their large plaques. Now, this finding means that the initial virus particle which caused this large plaque, this variant plaque, must have been a hereditary variant or mutant. A mutant which was changed in some characteristic which determined the way in which it grows on bacteria, and which characteristic ultimately manifests itself by the large size of this plaque. This original mutant particle must have passed on the large plaque-forming ability to all of its offspring, and is therefore a stable hereditary variant. Now, let's examine once more the role that the viral DNA plays in this reproductive process. You recall that it was the only component of the virus which entered the bacterial cell. Therefore, it must be the DNA which carries the hereditary message of the virus. It must be the substance which determines whether out of the bacterial cell come viruses of the small or of the large plaque type. Now, where do these hereditary variants or mutants come from? How do they arise? Well, they must arise as copy errors during the reproduction process. Now, let us imagine that the original DNA of one of the virus particles has entered the cell, and we'll represent it now as a straight line. Now, when this DNA reproduces itself, it reproduces itself in general with high fidelity. It makes an exact and faithful copy of the original parental DNA molecule. But once in every 10,000 or so times, something goes wrong in the copy process. Here, we're making a faithful copy, but at some place, an error occurs, so that now the message has not been copied correctly. We then continue and complete a DNA molecule, which is like the parental DNA molecule in every respect, except that at one place there is something wrong, something is different. Now, when this mutant or variant DNA molecule reproduces itself, it will make also one of these mutant or changed molecules. Now, a virus particle which manages to draw one of these mutant DNA molecules as its genetic material will be then a mutant virus and will pass on to all of its descendants this mutant property. Now, we've seen one way in which new virus types can arise, and that is by the copy error or mutation process. There exists, however, a second way in which new virus variants can be produced, and that is by the process of genetic recombination. Now, it may surprise you to learn that even organisms as small as these bacterial viruses have something like a sexual process. In the experiments we've considered until now, a bacterial cell has always been infected by a single virus, and the offspring viruses were always the descendants of just that single parent virus. But it is also possible that two virus particles can simultaneously infect the same bacterial cell. And in that case, it is found that among the offspring of these two virus particles, there will be some which have derived some of their genetic properties from one and some of their properties from the other of the two parents. Now, before we can consider the experiments which demonstrate such genetic recombination process, such sexual processes among the viruses, we must consider first another type of mutant. Now, if we look again at thousands of plaques, we will find another variant plaque, the turbid type, which we can see in close-up in the next slide. Here we see the turbid plaque more closely, and you see it differs from the clear plaque very clearly. Now let's review the three plaque types with which we have become familiar until now. There was, first of all, the small clear type, from which we started our discussion. And then we isolated the first mutant, which, as you recall, was large and clear. And then we isolated the second mutant, which, you remember, was small but turbid. So that we are now in the possession of three hereditarily different virus types: the small clear type, the large clear type, and the small turbid type. Now let us infect a bacterial cell, not with just one parent, but let us infect the bacterial cell with one virus each of the large clear and the small turbid variety. In other words, we are making what amounts to a cross, a genetic cross of two virus types. We then incubate these virus-infected bacteria for the usual 30 minutes. At the end of 30 minutes, several hundred offspring viruses will emerge from the cell, and we then put these offspring viruses on a jelly surface and see what kind of plaques do these viruses make. And then we find the following result. We see that on this plate there do not appear two, but there appear four distinct plaque types. Here we see the large clear plaque, and here we see the small turbid plaque, the two parental plaque types which entered this cross. But we also see two other types on this plate. We see, first of all, the small clear type, but we also see a type which we have never seen before, namely a large turbid plaque type. Now these two types, the small and clear and the large and turbid, which have appeared among the offspring of this cross, must represent genetic recombinants. For instance, the small clear type must have derived its small character from this parent, and it must have derived its clear character from this parent. Similarly, the large turbid one must have obtained the large character from this parent and the turbid character from this parent. Now, how do these genetic recombinants arise in the process of intracellular virus growth? They arise by a process which has been called copy choice. Now, in order to understand the nature of the copy choice process, we must visualize the genetic structure, that is to say, the DNA of these viruses in some symbolic form. And we will represent the genetic structure as a straight line. Now, the first of the two parents, the large clear parent, can be represented in this way. Somewhere here along the DNA must be some marker, some characteristic which spells out the large character. And somewhere else must be another site at which the DNA carries the information for the clear character. So this then is the symbolic representation of the first of the two parents. The second of the two parents, the small turbid one, can be represented by a second straight line, where at this corresponding locus we now have the information for the small character. And similarly, at this site, there must be the information for the turbid character. These are then the two parental DNA types which find themselves within the same bacterial cell at the outset of the infection. Now let us imagine that one of the two parental DNA molecules serves as a template for the reproduction of one of its offspring types. So we begin to copy this parent DNA molecule for a while and incorporate the large character into the replica molecule. But if somewhere within that mixedly infected cell, the second type of DNA is in the neighborhood, then the possibility exists that the copy process continues not along the DNA molecule along which it has started, but that a switch or a copy choice takes place so that the copy process is continued and completed along the other of the two parents, giving rise to a segment on which now the turbid marker has been copied. And you see now that the DNA molecule, which has been constructed by this copy choice process, carries the large marker at this side and the turbid marker on this side. It is therefore the large turbid genetic recombinant which we had seen to appear from this cross. Now let us summarize the knowledge which has been gained from our study of bacterial viruses. We saw, first of all, that it is only the viral DNA which enters the bacterial cell. It, therefore, must be the hereditary material of these viruses. The viral DNA must be capable of reproducing itself several hundredfold inside the infected cell, and it must be able to preside over or induce the synthesis of the viral protein. Secondly, we saw that in the course of this self-duplication of the viral DNA, copy errors can occur, copy errors which give rise to new or mutant virus types. And lastly, we saw that if two bacterial viruses, which differ from each other in their hereditary properties, infect the same bacterial cell, then recombinant viruses can emerge from that cell, which have derived some of their genetic properties of one and some of their properties from the other of the two parents. Now, this knowledge which we have gained is valuable from 2 points of view. First of all, it has helped us to understand the nature and function of the genetic material, that most fundamental substance of all living things. And secondly, it has aided us in our efforts to control other viral diseases, such as those caused by the polio or by the tobacco mosaic virus.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program was Dr. Gunther S. Stent, Professor of Virology. This series was made possible by a grant from the National Science Foundation. This is National Educational Television.